Hi Rachel, I'm coming to you today from a room of dim lighting because this brought to you by the fact that the sun sets at like 2 in the afternoon. So you'll have to deal with the sun in my hallway and dim lighting in my room. We're just going to get that out of the way right now. So without further ado, let's start with book news. Okay, book news is really boring. So I hyped that up for no reason. So book news is I'm still reading The Secret of Christopher Topher. I can only read like 10 pages of that book at a time. It's really bad. Can't do anymore. So that's going to take a while. I'm starting with Lady Thy Name is Trouble tomorrow while I'm on the bus because tomorrow is Thanksgiving. Happy Thanksgiving. So I thought about doing a video about the history of Thanksgiving for this week, but no. I didn't want to relive elementary school history class. I didn't think you did either. So I decided to do a topic completely not related to anything with the holidays at all. I'm going to talk to you today about PCR. Whoa! Coolest thing ever! Woo! Favorite lab technique. So you might be wondering what PCR is. So PCR stands for polymerase chain reaction. Why would I want to use PCR? That might be your next question, and that name doesn't even make sense. So what does it do? So basically PCR amplifies a target segment of DNA. So if you have one sample of DNA and you want a lot of a small sequence of the DNA, you use PCR to amplify it and make a bunch of it so you can run an analysis, like running a gel, to get a visual look at the size of your DNA or protein or RNA or whatever you want. So what do you need to do to start PCR? You might be wondering now because it sounds like a fun time. Everyone wants lots of DNA. I don't know who wouldn't. So to start off, you're going to need a machine called a thermocycler. Ooh. A thermocycler is a machine that, as the name implies, cycles temperatures. Ooh. So what it's going to do is, a vital part of PCR is moving the temperature from very hot to cooler to like moderately warmer to make the process go along smoothly, which will make more sense once I explain the rest of the process. So then you're also going to need Taq polymerase. Whoosh. Taq polymerase. Taq polymerase is, you might have heard of DNA polymerases before. Well, you can't just use any ordinary run-of-the-mill DNA polymerase because DNA polymerases have this super unfortunate side effect of denaturing at high temperatures, and since we're going to be exposing our target DNA to high temperatures and therefore our polymerase to high temperatures, it would become useless. So we use Taq polymerase because Taq polymerase is found in thermal vents, so it doesn't care about heat. It's like 95 degrees Celsius? Alright, that sounds fun. So also you're going to need to throw some magnesium chloride buffer in there just to keep every pH all happy. You're going to need some primers. So you have primers for your target DNA sequence, which is very important so that the polymerase will have something to sit on and start moving along. And you're going to need some bonus nucleotides to throw in there so you have the parts to make your other strand of DNA. And of course, you're going to need your target DNA sample, which, why wouldn't you put that in there? So to get started, there are 3 steps. Super easy. The 3 steps just keep repeating over and over and over again. And you don't really have to do anything. The machine does everything for you. But we can take a look at what's going on inside. I have a marker and a whiteboard. I'm official. Alright, so your first step is the denature step. No, my marker's dying. I'm not that official. Okay. No. Okay. Denature. So what's happening in the denature step? Your little test tube full of all the ingredients that we just put in gets plopped into the thermocycler and starts off by denaturing your DNA. So let's see, this is your target DNA. We have your little lovely target DNA hanging out in your test tube. You raise the temperature to 95 degrees Celsius. So this is super hot, and the bonds between the nucleotides aren't going to want to stick around with that at that high temperature. They don't want to touch. because it's too hot for touching. So your strands will break apart and become 2 single strands of DNA instead of 1 double strand piece of DNA. And that's exactly what we want for our second step, which is the anneal step. So this is done, so then after we've denatured our DNA, the thermocycler is going to drop the temperature down to about 55 to 65 degrees Celsius. The exact temperature is going to vary depending on what DNA you're using, what project you're doing. Just vary, but it's going to be in there approximately. So what this step does is we have our 2 separate strands of DNA now. The anneal step is going to let our primer attach to each of the single strands. So it's going to sit on there. The reason we want to drop the temperature is because if we left it at this super high temperature, the primers would not anneal to the single strands of DNA. They would break apart. They'd be like, I'm out of here. It's too hot. So we have to drop the temperature. Then, our third step is elongation. So in the elongation step, we raise the temperature a little bit to about 72 degrees Celsius. It's not hot enough to make anyone break up with each other, but it's a little warmer, just get things moving along. This is when so we got our strands. We got our strands here, and we got our little primers. And then this is when our Taq polymerase that we put in earlier, that was swimming around just like the primers, comes in and it binds onto here, just like that. It's like, hey primer, what up? And it starts adding the nucleotides that we also added into the solution, just like putting the little bricks together. And it's going to move in the 5' to 3' direction, just like all DNA synthesis does. So then this guy goes this way. And then we end up with 2 sets of double-stranded target DNA. So then we have 2. That's great, but we want more than 2. So the cycle repeats. These guys are both denatured, primers anneal, they elongate, and then we have more. And it just keeps going up and up and up and up. It depends on how much you want from this. You can run it hundreds of times if you really wanted to. Nothing better to do. Or a couple, just depends on how much you need. So that's PCR. So that about does it for this week. I hope you enjoyed this video, and I can't wait to see yours. Bye!